Hey everyone, welcome back. Today, we are cracking open question 8 from the 2025 VCE Biology Exam, a massive 14-mark question worth over 11% of your entire Section B. We are looking at the wild experiment where scientists injected solar-powered bacteria into the hearts of tadpoles to literally restart flatlined brain activity. The science sounds like a movie, but the marking scheme was a minefield. Thousands of students dropped easy marks by falling into classic traps on photosynthesis, respiration, and bioethics. In this quick walkthrough, I'm going to show you the exact trap avoidance secrets from the Chief Examiner's report so you can lock in a perfect 14 out of 14. Let's get straight into it. Scientists are using microorganisms to restart brain activity in tadpoles. Animal brains are made up of specialized cells called neurons. The activity of these cells relies on a constant supply of oxygen. When oxygen supply is too low, hypoxia, brain activity stops. Scientists often use tadpoles during research. Tadpoles hatch from eggs that are laid in groups of thousands. Their skin can be transparent, as shown in the diagram below. During the early stages of their development, tadpoles absorb dissolved oxygen from the water through their gills. Researchers conducted an experiment using tadpoles. The experiment was approved by the Ethical Standards Committee. The tadpoles were used to explore whether photosynthetic cyanobacteria could serve as an alternative oxygen source to that supplied in the water. Researchers injected cyanobacteria into the hearts of tadpoles. The cyanobacteria entered each tadpole's bloodstream and were transported to other parts of the body, including the brain. After the injection, these tadpoles were placed in a dark, hypoxic environment. After about 65 minutes, brain activity stopped, indicated by the white arrowheads in the figure below. These tadpoles were then exposed to light. After 15 to 20 minutes in the light, brain activity restarted, indicated by the black arrowheads in the figure below. In the future, this might provide a way to increase oxygen levels to humans during medical emergencies. However, a major challenge for a future use of injectable photosynthetic microorganisms is the potential activation of an immune response in people. Much more research is needed to ensure the safety of this potential treatment. Using information from the article, propose 2 reasons why tadpoles were chosen for this experiment. Their skin can be transparent. Allows light to penetrate the body tissue, so the photosynthetic cyanobacteria can absorb it to produce oxygen. Eggs that are laid in groups of thousands. Provides a large sample size easily, increasing the reliability of the experimental data. Approved by the Ethical Standards Committee. Tadpoles are less complex animals, meaning invasive procedures, like cardiac injection, face fewer ethical barriers. Absorb dissolved oxygen from the water through their gills. Scientists can easily manipulate the oxygen levels in the surrounding water to induce a hypoxic, low oxygen, state. Tadpoles are small animals. They are highly cost-effective and logistically easy to house and maintain in a laboratory setting. While any 2 reasons from the classification matrix will satisfy the criteria for the full 2 marks, the following examples demonstrate how to pair the most relevant facts from the text with their corresponding experimental design reasons. Example 1. Combining transparent skin plus large sample size. Reason 1. The text states that tadpoles can have transparent skin, which allows light to easily pass through their bodies to be absorbed by the cyanobacteria, so they can produce oxygen. Reason 2. The article mentions that tadpoles hatch from eggs laid in groups of thousands, which enables researchers to easily gather a large sample size to ensure more reliable data. Example 2. Combining transparent skin plus ethical approval. Reason 1. Tadpoles have transparent skin, which ensures that when they are exposed to light, the light can actually penetrate the tissue to stimulate photosynthesis in the injected microorganisms. Reason 2. The article notes that the study received ethical approval, which is because tadpoles are less complex animals, making it easier to justify invasive procedures like injecting bacteria into the bloodstream. Example 3. Combining environmental control plus large sample size. Reason 1. Tadpoles live in water and use gills to absorb dissolved oxygen, meaning that scientists can easily control and lower the oxygen levels in their water to create the hypoxic environment needed for the experiment. Reason 2. The text implies tadpoles hatch from eggs laid in thousands, making it highly accessible for researchers to obtain a large sample size for a more reliable test. B.I. Other than enzymes, name 2 molecules the photosynthetic cyanobacteria would require to produce oxygen. 2 marks. Pay close attention to the constraints in this question. It explicitly rules out enzymes, and specifically asks what molecules are required to produce oxygen. Many students reflexively write down carbon dioxide or light when they see photosynthesis, which are classic traps where marks are lost. The key to tackling this question is to exclude enzymes entirely and answer using 2 fundamental small molecules directly involved in the light-dependent stage. Core teaching linkage. Ideal scoring combination: water, H2O, and chlorophyll. The logical chain. Chlorophyll molecules absorb light energy drives the photolysis of water molecules directly releasing oxygen gas. This entire process perfectly avoids enzymes and relies strictly on valid chemical molecules. Other accepted answers are coenzymes, NADP, or ADP plus pi. Student Trap Avoidance Guide. To secure the full 2 marks, students must actively steer clear of these 4 common mistakes highlighted in the official examiner comments. Crossmark Trap 1, the Calvin cycle reflex, carbon dioxide or glucose. The mistake, writing CO2 or glucose. Why it loses marks. These are inputs and outputs of the light-independent stage, Calvin cycle. They have no direct biochemical role in the splitting of water or the generation of oxygen gas. Crossmark Trap 2, category mistake, light. The mistake, writing light or light energy. Why it loses marks. While light is absolutely required to drive photosynthesis, light is not a molecule, it is a form of electromagnetic radiation. The question explicitly demands molecules. Crossmark Trap 3, ignoring question constraints, enzymes. The mistake, writing rubisco, ATP synthase, or general enzymes. Why it loses marks. The question stems strictly explicitly stated, other than enzymes. Any mention of an enzyme immediately invalidates that point. Crossmark Trap 4, generalizing photosynthesis. The mistake, just reciting the overall photosynthesis equation without isolating the light-dependent stage. Why it loses marks. Students must narrow their focus exclusively to the reactions occurring at the thylakoid membrane, where oxygen is actually manufactured. Moving into Part B.2, the examiner is looking for a functional description of the exact 2 molecules you just wrote down. The key here is functional alignment. Your explanation must directly target how that specific molecule contributes to the production of oxygen. Let's look at how to match your descriptions perfectly to your Part BI answers to lock in the next 2 marks. Ideal student response examples, based on Part B.I choices. Depending on the 2 molecules your students selected in the previous part, they should provide the corresponding pair of descriptions below. Pairing A, if the student chose water and chlorophyll, are highly recommended. Role of water. Water is split during photolysis to directly form oxygen gas, while also providing electrons and protons to load coenzymes like NADP+. Role of chlorophyll. Chlorophyll captures and absorbs light energy, which is required to facilitate the splitting of water and the loading of coenzymes. Pairing B, if the student chose water and coenzymes. Role of water. Water acts as the initial reactant that is split to generate oxygen gas and load empty coenzymes. Role of coenzymes, e.g., NADP+, the ADP plus pi coenzymes accept and transfer electrons, energy, or hydrogen ions between reactions, assisting enzymes to function during the process. The consequential marking lifeline. Warning sign # the light consequential mark exception. If a student made the classic mistake in Part BI of listing light as a molecule, they would receive 0 marks for that specific point in BI. However, if they proceed into Part B.2 and correctly describe the role of light, stating that light energy is captured to split water and produce oxygen, the examiners will award 1 mark for that description in Part B.2. What this looks like in practice. Part B.I, student writes, 1. Water, 2. Light. Scores 1/2 marks, light is not a molecule. Part B.2, student writes. Role of water, it is split to form oxygen. Scores 1 mark. Role of light, it provides the energy to split water to produce oxygen. Scores 1 mark, consequential mark saved. Tips, never leave Part B.2 blank, even if they feel uncertain about what they wrote in Part BI. The marking guide contains pathways to rescue dropped marks. When a question asks you to explain why brain activity stops in a hypoxic environment, you cannot just say, because there is no oxygen. The examiners want to see the underlying biochemical cascade. This is a classic cellular respiration logic chain question. You need to link a lack of an environmental input directly to a failure in cellular energy production, and finally to a failure in organ function. The 2-mark breakdown, the logic ladder. To secure the full 2 marks, a student's response must cover 2 sequential logical steps. Mark 1, the cellular impact state that a hypoxic, low oxygen, environment limits or prevents aerobic cellular respiration in neurons, drastically reducing the production of ATP, cellular energy. Mark 2, the physiological result explain that neurons require a constant, high supply of ATP to function, e.g., maintaining membrane potentials, transmitting nerve impulses. Without it, they cannot fire, causing brain activity to cease. Ideal student response examples. Example 1, direct and technical, highly recommended. In a hypoxic environment, the lack of oxygen prevents neurons from undergoing aerobic cellular respiration, which severely reduces the production of ATP. Because neurons require a constant supply of ATP to transmit nerve impulses and maintain basic cellular functions, the lack of energy causes brain activity to stop. Example 2, clear cause and effect. 1. Hypoxia means there's insufficient oxygen available to act as the final electron acceptor in aerobic respiration, preventing the efficient generation of ATP. 2. Without adequate ATP, the tadpole's neurons lose the energy required to fire electrical signals, leading to the cessation of brain activity. Student Trap Avoidance Guide. Crossmark Trap 1, vague terminology, energy. The mistake, saying the cells run out of energy. Why it loses marks, energy is too broad for VCE biology. Students must explicitly name the molecular currency of the cell, ATP. Crossmark Trap 2, skipping the cellular link. The mistake, saying brain activity stops because neurons need oxygen to live. Why it loses marks. This jumps straight from the environment to the symptom, skipping the whole reason why they need oxygen. They must explicitly mention that oxygen is required for aerobic respiration. This question addresses the 15 to 20 minute time gap observed between turning on the light and the actual resumption of neural firing. To secure the full 2 marks for the question, we must construct a logical bridge that explicitly links a named biochemical pathway to a delayed physical outcome. When explaining the delay in brain activity, the biggest trap is saying something vague like, "It takes time for things to start working." The prompt explicitly demands that you refer to a biochemical pathway. To score both marks, you need to state which pathway takes time to progress, such as photosynthesis or cellular respiration, and connect that time lag directly to the eventual production of oxygen, or ATP, required by the brain. The 2-mark breakdown, the logic ladder. Mark 1, the pathway delay. Identify a specific biochemical pathway, e.g., photosynthesis, light-dependent stage or aerobic cellular respiration, and state that it is a multi-step process that takes time to occur and build up products. Mark 2, the physiological link. Connect the output of that delayed pathway directly to what the brain needs to restart functioning, oxygen, or ATP. Ideal student response examples. You can teach your students 2 different angles to answer this question perfectly. Approach 1, focusing on photosynthesis, the production delay. When exposed to light, it takes time for the photosynthesis pathway to occur within the cyanobacteria. Specifically, the process of photolysis takes time to split water and generate enough dissolved oxygen to diffuse into the bloodstream and reach the brain. Approach 2, focusing on cellular respiration, the energy delay. Once oxygen is available, it takes time for the tadpole's neurons to complete the multi-step pathway of aerobic cellular respiration. This biological delay means ATP cannot be generated instantly, and a sufficient threshold of ATP must accumulate before the neurons have enough energy to restart brain activity. Student Trap Avoidance Guide. Crossmark Trap 1, mere identification, no connection. The mistake, writing, the delay is because photosynthesis and cellular respiration take a long time to happen. Why it loses marks. According to the official comments, simply naming a pathway without linking its metabolic lag to the final physiological outcome, brain activity ATP needs, will not score full marks. Crossmark Trap 2, skipping the time element. The mistake, writing, photosynthesis produces oxygen, and then cellular respiration produces ATP for the brain. Why it loses marks. This answers how it happens, but forgets what the question asked. The response must explicitly address the delay by using time-oriented phrasing, such as "it takes time to accumulate sufficient levels," is a slow, multi-step process. When a VCE biology question asks you to outline events along the electron transport chain, etc., you don't need to get bogged down in complex university-level biochemistry mechanisms. According to the VCAA study design, the key is knowing your core inputs, outputs, and energy yields. Let's check out how to state 2 distinct, straightforward events along the CRIS tie to pick up these 2 marks efficiently. Accepted events, choose any 2. To achieve the full 2 marks, a student must outline 2 clear, separate events that happen during this mitochondrial stage. Coenzyme unloading. High-energy coenzymes, NADH and FADH2, transfer and unload their electrons, hydrogen ions, plus, or energy down the chain. Oxygen usage. Oxygen acts as an input, the final electron acceptor, at the end of the chain. Water formation. Oxygen binds with electrons and hydrogen ions, resulting in the formation of water as an output. Mass ATP production. A large yield of energy, specifically 26 or 28 ATP molecules, is produced via phosphorylation. Ideal student response examples. Example 1, focusing on inputs and outputs, simplest method. Event 1, oxygen is used as a terminal input at the end of the electron transport chain. Event 2, water is formed and released as a major product of this stage. Example 2, focusing on energy and coenzymes. Event 1, loaded coenzymes like NADH and FADH2 unload their electrons and hydrogen ions into the transport chain complexes. Event 2, a large amount of cellular energy is captured, producing 26 or 28 ATP molecules. Student Trap Avoidance Guide. Trap 1, getting caught in over-complex mechanisms. The mistake, spending time explaining proton pumps, electrochemical gradients, or the rotational mechanism of ATP synthase. Why it loses efficiency. The examiner notes state explicitly that details of biochemical pathway mechanisms are not required. Keep responses limited to the big picture inputs, outputs, and coenzyme states to save valuable exam time. Trap 2, generalizing ATP yields. The mistake, writing a lot of ATP is made, or 30 plus ATP is produced. Why it loses marks. VCE specifications require exact numbers for specific stages. For the etc. alone, you must explicitly declare either 26 or 28 ATP to be technically accurate. Avoid using old textbook numbers like 32 or 34. Trap 3, confusing locations. The mistake, mentioning the conversion of pyruvate into acetyl-CoA or the release of carbon dioxide. Why it loses marks. Those events belong to the link reaction and the Krebs cycle occurring in the mitochondrial matrix. The question strictly asks for events occurring along the chain itself, the inner membrane of cristae. Question F is a brilliant example of a VCE bioethics question, where there is no single correct answer. The examiner report shows a very high success rate, 69% scoring full marks, because it is absolutely not necessary to select both student X and student Y. You only need to choose one student. The full 2 marks are awarded entirely based on how well you define your single chosen principle and back it up with facts from the article. Student Trap Avoidance Guide. Crossmark Trap 1, the sitting on the fence mistake, trying to select both. The mistake, writing about both student X and student Y to try and cover all bases, such as I agree with both because. Why it loses marks a time, the prompt explicitly instructs you to state which student you agree with singular. Trying to argue both sides splits your focus, makes your justification messy, and risks running out of space. Pick one side and defend it beautifully. Crossmark Trap 2, the principal-student mismatch. The mistake, writing, I agree with student X because the experiment caused harm to the tadpoles by stopping their brain activity. Why it loses marks. The official notes state that some responses failed because their justifications did not match the student they selected. If you choose student X, you must discuss beneficence, maximizing benefits for saving human lives. If you choose student Y, you must discuss non-maleficence, avoiding harm or distress to the tadpoles. Crossmark Trap 3, omitting the article's facts. The mistake, giving a purely textbook definition like, I agree with student X because beneficence means doing good things. Why it loses marks. The prompt explicitly demands that you justify your response using information from the article. You must tie the bioethical definition directly to the text, such as mentioning the potential to save human lives in medical emergencies, or the fact that the tadpole's brain activity safely restarted. Ideal student response examples. Option A, choosing student X, Beneficence. I agree with student X. Beneficence involves maximizing positive outcomes and benefits while minimizing harm. This experiment aligns with this principle because the research aims to maximize a significant positive outcome, potentially saving human lives during medical emergencies by finding alternative oxygen sources, while the harm to the tadpoles was minimized since their brain activity was successfully restarted. Option B, choosing student Y, Non-maleficence. I agree with student Y. Non-maleficence involves a commitment to avoiding harm or ensuring that unavoidable harm is not disproportionate to the benefits. This experiment can be viewed as unethical under this principle because it inflicts direct, intentional harm and distress on living organisms by injecting bacteria into their hearts and forcing them into a hypoxic state where brain activity stops. Q8a Choice of tadpoles. Transparent skin lets light reach injected cyanobacteria, and eggs laid in thousands give a large sample size for reliable data. Q8b molecules for oxygen production, water, HO, and chlorophyll avoid glucose, light, or enzymes. Q8b.2 roles. Chlorophyll absorbs light energy that drives photolysis of water, releasing O and loading NADP. Q8c Why brain activity stopped. Hypoxia blocks aerobic respiration in neurons, reducing ATP needed to fire nerve impulses. Q8d Restart delay. 15 to 20 minutes photosynthesis and aerobic respiration are multi-step pathways. Time is needed to accumulate enough O and ATP for neurons to restart. Q8e Electron transport chain. Coenzymes NADH, FADH, unload electrons but H. O is the final electron acceptor forming water, yields 26 or 28 ATP. Q8f Bioethics. Pick one student, justify with article facts. Beneficence, student X, equals potential to save human lives. Non-maleficence, student Y, equals harm or distress from cardiac injection and induced hypoxia. Exam tip, use precise terms, ATP, photolysis, aerobic respiration, respect question constraints. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.